Hello, my name is Brandon Tonnis. Seed trait analysis is an important part of many plant breeding programs. For oilseed crops like peanuts, oil content and fatty acid composition are often identified as traits of interest. In this video, I will explain how to do fatty acid analysis on peanut seeds using gas chromatography. The analysis can be broken down into 4 steps: sampling the seed tissue, extracting the oil, converting the fatty acids into a measurable form, and then finally measuring them by gas chromatography. To sample the seed tissue, a small amount is cut from the distal end away from the embryo. In this way, the seed remains viable in case germination is required later. Only a small amount is needed, about 50 milligrams or so. It is placed into a glass tube and then ground using a disposable plastic tip. A separate tip is used for each sample, and the razor blade is cleaned between samples using alcohol to prevent cross-contamination. Oil is extracted from the seed tissue using an organic solvent such as heptane. This step and subsequent prep steps are done in the fume hood to avoid exposure to the chemical vapors. At the same time, methanol and a base catalyst are added to chemically convert the oil into individual fatty acids and modify them to produce fatty acid methyl esters, or FAMs. The tubes are then capped and vortexed to mix the chemical reactants. After the reaction is complete, the caps are removed from the tubes and water is added to each. This will allow 2 layers to form: a top layer of heptane containing FAMs and a bottom layer containing water and the residual seed material. After about 45 minutes, the 2 layers have separated and the top heptane layer is completely clear. Sampling only from the top layer, approximately 1 milliliter is taken and aliquoted into the injection vial. The vials are capped and sealed and are now ready for injection onto the gas chromatograph. Gas chromatography uses an inert gas such as helium to carry the injected sample through a column, where the individual components of the sample are separated using heat. They are then detected and measured. Compounds must be both volatile and thermally stable, which is why the oil is converted into FAMs. The vials are placed in the tray and the autosampler arm automatically loads the selected sample. Up to 100 vials can be added to the tray at a single time. The injection needle is washed to eliminate contamination from previous samples. Then the selected sample is injected onto the column. The accompanying software measures in real time each fatty acid as it is detected by the instrument. The compounds are represented as peaks, and the height and area of each peak is proportional to the relative amount of the fatty acid in the oil sample. The end result is called a chromatogram, which shows each individual fatty acid and its relative concentration. While there are other methods for estimating fatty acid composition, FAM analysis using gas chromatography is an accurate and efficient method for screening large numbers of samples in a breeding program.